and I'm happy to present you about learning from trees and how we models simulate and infer microbial genome evolution. So my background is in mathematical population genetics, and what we are mainly interested in, if I wanted to explain it in a short sentence, is we're trying to explain natural variations within and between species. And to do so, we are building stochastic evolutionary models. We are estimating parameters of the evolutionary models. We try to identify elements under selection, infer ancestral relationships, and also related is predicting population dynamics, maybe spread of disease. Some of my colleagues are working on that, but I haven't been working with COVID so far. So trees are very important in population genetics, just because if we look at the pattern we observe, which emerge by the reproduction of individuals, then it's natural that trees occur. So here's the White-Fisher model shown, where at the present, each dot represents an individual, and then each individual chooses a parent of the former generation randomly, and then we trace back the parents of those, so the ancestors, by tracing back those. And at some point, two individuals choose the same parent. And here we have two individuals which choose the same parent, and they both have been sampled in or marked in blue here. And then if we look at the tree connecting those three individuals here, then we'll have a merging event at this point here. So these two dots choosing this dot represent this merging event here in the tree. So it's natural that trees occur in population genetics if we look at evolution. However, it's of course more complicated than just dots choosing parents. Here's a major difference between human population structures and evolution and microbial evolution. So for humans, we usually assume that we have strong human population structures. And in addition, we are diploids. So each of us has two parents, and each of our chromosomes comes from a different one. But even along the same chromosome, at some point, we have recombination events, which means although it's one chromosome, its origin has two different parents. And by that, we get a different structure. In contrast, for bacteria, we usually have a much more clonal reproduction for bacteria, as they just reproduce by dividing into two new cells, which are usually identical to the parent. And of course, there's some more and other features of microbial evolution, which I'll come to, but this is really a major difference, making trees even more important for microbial evolution. And the reason is the following. If we are looking at evolution of a chromosome in a human population, then along that chromosome at some point, you'll have recombination events where both parts of the chromosome, for example, here at the point B2 along the chromosome, we have two different parents which occur here. You can also see that in, so if this is the chromosome here from B equals 0 to B equals L, then at B2, the tree which connects the local ancestry of this part of the genome changes to this tree as now a different structure. So now 1 and 2 are closer related to 3 than 3, and before 3 and 4 have been closer related to each other. And this leads to a sequence along the chromosome, which we call the ancestral recombination graph, where it's a sequence of dependent trees. And we've been able to look at the same process in bacteria. So in bacteria, usually you just have clonal reproduction, but from time to time, there's gene transfer between different individuals. And at that point, only a small fraction of the genome is exchanged between ancestors. And we were able to show that this structure can be represented by another graph, which we call the ancestral gene transfer graph. And in this graph shown here, we have one clonal tree, which is very important. And then all the other trees depend on each other, but always also on the clonal tree. So clonal trees are important. And as Philip already mentioned, I'm also part of the I'm a member of the cluster CMFI, and there we are looking at microbiomes. But also for microbiomes, it's really important to look at the diversity within individual strains of your bacteria. And the evolution of microbial organisms living in our microbiome and our own evolution is, of course, also entangled. So the population structure of humans can, or sometimes does not, but often can also affect the population structure of bacteria. And we are trying to look at, in our group, we are trying to look mostly at the variation within species or one species and looking at different strains, and not so much looking at diversity between species. And one thing we looked at, and which is really different from human evolution as well in bacteria, is the distributed pangenome of bacterial populations. So a pangenome describes the total set of all genes in the population. So here's a chart where if you'd sampled more and more human genomes, how many genes would you find? And for human genomes, that would just be a constant number. Of course, you find single point mutations and variations of the same gene, but basically all humans have the very same genes. In contrast, if you look at the E. coli genome, then for each new E. coli cell or strain you sequence, you'll find a few more new genes you haven't observed before. And this diversity and this distributed genome is called the pangenome in bacterial populations. And we've been developing models which describe this evolution. So we call this the infinitely many genes model. And this provides a mathematical framework for this pangenome evolution. And in this model, what we do is we can gain genes along a clonal tree. We can lose these genes again, but each gene we gain is always a new one. So losing it means we won't be able to recover the same gene. And then this dynamics leads to a gene presence absence matrix, which describes all the genes present or absent in each of the individuals. And the individuals are connected by this tree. And then we were able to show things like, if we know this underlying tree, then we can compute the underlying distribution of gene frequencies in the population. So here, the number of genes which are present in K, any K out of N individuals is Poisson distributed with some parameter lambda, which depends on the gene gain rate, the gene loss rate, and the tree. And we can compute this. However, this data structure in population genetics means we have these huge matrices, which are basically zeros and ones. And you describe either whether you have a gene or you do not have a gene, or whether you have a mutated site at that position or not. And it's turned out that we can do much better in encoding this whole information if we use a tree-based encoding, which Jerome Kellerer has developed in this publication here. And by doing that, so by instead of encoding the whole matrix, we now have two tables, one describing the tree and the other one describing variants or mutations, we can dramatically decrease the size and the number of operations we need to do in simulations and the size of the data we generate. So for example, if you would like to encode the complete human population, you can do so in the new in this tree sequence format below 1 terabyte of data if you compress it even more. So this is an amazing improvement, and also the simulation speed is really improved much. We've also computed from the theoretical results, created some data, some biological software for pangenomes. One is Pangenome, which can estimate the gene gain and loss rates, and the other one is called PanX, which I've done together with Reweighting and Richard Neer in Tübingen a while back ago. And PanX can just takes all the bacterial genomes, clusters the genes into groups of orthologous genes, estimates clonal species trees and all the individual gene trees, and then computes ancestral gene gain and loss events, and then also, due to mainly the work of Reweighting, can beautifully, very beautiful, illustrate everything on an interactive website where you then can browse the pangenome. And this is a screenshot of this website. And you can, here is an example where you can show, you can have a look at the species tree, which I already mentioned in the ancestral gene transfer graph. So this is the gene, the tree we think is closest to the clonal reproduction of the bacteria. And here on the other side is a gene tree, which represents the genealogical history of one individual gene. And then we can use this information of all these ancestral events along the trees to associate the presence and absence of genes to phenotypic data. Here's an example I did together with Fabian Staubach in Freiburg, where they measured the offspring of fly, which has been, and those fly have been colonized with different strain variants of Gluconobacter in their microbiome. And then some of them had rather few offspring, and we were able, using this association method based on the events on the tree, to associate the thiamine pathway, so thiamine is vitamin D, to this to the number of fly offspring. So the white organisms here, they lack the thiamine pathway, so they can provide vitamin D to the flies. And then if you look at, and then you can browse in PanX, and then you can look at the same data here, and then you see those two guys here are the farthest related in the species tree, but the gene tree shows that those guys here have regained it. So what we think has happened is, so this is the species tree, this is the gene tree on the right, and this blue part here is the new genes which have been coming in by horizontal gene transfer. And you can see that as those two guys here are closer related to all the other ones as to these ones. So what we think has happened is it's been lost here and then regained here by horizontal gene transfer. And all of this can be reconstructed from those tree information. So our goal is now to sort of use the same ideas that all the information in the trees is really important in population genetics to develop new and understand machine learning approaches in population genetics. And in the last years, there have been some progress in this area. Here are three, in my opinion, outstanding publications for machine learning in population genetics in the last years. This upmost here by Flegel has the beautiful name, The Unreasonable Effectiveness of Convolutional Neural Networks in Population Genetic Inference. And what they did is they supported a convolutional neural network with the with a mutation presence absence matrix. So basically interpreting this matrix as a picture and just giving it to a convolutional neural network. And that's why they call it the unreasonable effectiveness, as we usually do not think of site mutation data tables as pictures. And then, and one example is, and one thing is, if you resort the data, so if you reshuffle the position of the individuals, you usually wouldn't expect a different result of your estimation methods. But the convolutional neural network, of course, in this study at least, depends on this sorting and produces different results. And thus, in this study here, an exchangeable neural network framework is suggested, where the exchangeability of neural networks is really important for population genetic data, as we usually do not rely on that. And then here, the group of Laura J. in Paris is really doing some very cool stuff, combining approximate Bayesian computation and deep learning in population genetics. So the main challenges for machine learning in population genetics are the following. We can mainly only create enough independent training data by simulations. And the reason is just, again, the tree which connects all human life, for example. So if you want to learn something about features of the human population, you would need multiple samples of human populations, and we only have one, which makes it hard to create independent training data. So that means we depend on training data, and then this training data is simulated, which means our models will depend on the model assumptions we do in this simulated training data. In addition, we have a rich feature set of classic tools and results which compete with current machine learning methods, which means we really need superior performance to convince people to switch the methods. However, there are some promising results in recent publications. And one of the biggest advantages, in my opinion, is that machine learning methods will be able to handle much larger sample sizes and data sets, which is often a problem in classical approaches. So still, the main questions are, in my opinion, when should we trust in model-based methods, and when is machine learning a promising approach? And one thing, so the basics to get to this will be to effectively simulate large data sets in complicated or at least more involved evolutionary scenarios. And with standard PopSim and MS Prime, I'm part of two open source communities working on exactly that. So standard PopSim tries to create a standard benchmark resource for population genetics, which so far doesn't really exist. So everyone is simulating his own models and showing that his inference methods are working on his own models better than the other ones on the models of the other ones. And the second one is MS Prime, which is the super fast and efficient backward simulation based on these succinct tree sequences I've just shown a while ago. So I already have one PhD student in my lab or in my group, and this is Clara Burger. And Clara has been working on the connection of mutation rate estimators we already know from the theoretical literature, and tries to understand how machine learning algorithms do perform in this scenario where we know the theoretically best solution. So the scenario is as follows. We have this tree, and then along this tree at some rate, mutations occur, here shown as these crosses. So that means our data is either the side frequency spectrum, which describes how many mutations do I have which appear in one individual, which would be 1, 2, 3 here. How many mutations do I have which appear in 2 individuals? Those are these 2 mutations here, and this individual. And then there's 1 mutation which appears in 3 individuals. And then, of course, we could use a convolutional neural network and just give it the picture of the data. Or we can compute summary statistics, which is the usual approach to estimate those parameters. And here we are looking at the basic scenario where we're trying to just estimate this mutation rate theta, which puts these crosses along the tree. And if we restrict ourselves to the side frequency spectrum, so the number of mutations present in 1 up to n-1 individuals, and we're looking for estimators that are linear in this side frequency spectrum of this form here, then all we have to do is to identify those parameters AI to find the to find an estimator, and this estimator then can be optimized. So one example is Wattersons estimator, which just sets every A to the same value, which is 1 divided by the expected length of the tree, and then you basically take the total number of mutations and divide it by the expected length of such a tree, which gives you this estimator. And in this scenario, Fu 1994 showed or computed the coefficients that minimize the variance for all unbiased estimators based on the side frequency spectrum. And Fuchik and Gach in 2008 showed the calculated the coefficients that minimize the mean square error. And those are shown, so Fu is shown in blue here, and they differ. So the black ones are by Fuchik and Gach. And these coefficients here, they depend on theta. So the optimal parameter to estimate theta depends on theta. Of course, we do not know theta. So what we do in this case is we just approximate those estimators by iterative versions. So for example, we can start with Watterson's estimator, estimate some theta, then take the parameters which are optimal for that theta, and then proceed until this converges, which usually is really quick. So we were asking us, to what extent can even a minimalistic neural network already replicate or even outcompete these optimal estimators? So we created a very simple neural network with 1 hidden layer and trained it in this scenario. So simulated side frequency spectrums, and then we tried to minimize the normalized mean square training error. However, what we noticed at some point was that for some thetas, the performance was rather poor in comparison to the optimal. So, and we identified that this basically depends on the amount of data we assign to which part of the parameter space in the training data. So instead of doing this manually, we decided we include a reweighting, an adaptive training error, where we compare in each step the performance of model-based estimators and the neural networks, and where the neural network is still not close to the model-based estimator's performance, we increase the weight of the loss function in this area of the parameter space. And here are some first results. So everything, all of this is unpublished, but and has been done by Clara. So in the scenario where we have no recombination, we know that Fuchik has the lowest normalized mean square error. And then the adaptive neural network is shown in red and comes really close to these results. In contrast, the Watterson estimate has a higher variance, which is known from theoretical results. However, what about recombination? What if we include recombination? Because all of those optimal estimators by Fu and Fuchik have been computed in this scenario without recombination. And we know that in the limit of high recombination, Watterson's estimator is outcompeting the other ones and has the smallest variance, because if you have high recombination, then we have very many trees which are independent of each other due to the high recombination rate, and then dividing by the expected size of a random tree is a very good approach. And for intermediate recombination rates, it's really involved to compute those optimal estimators. So we asked, can the neural network estimators learn the optimal coefficients from simulated data, including recombination? And if we train them on data sets where we include recombination, we get very good results where the neural network performance is always close or sometimes a little bit better than the known optimal estimator. In this intermediate recombination rate regime, he's better than Fuchik and Watterson. And in this high recombination rate limit, he's close to Watterson's estimator performance. Then we went one step further and asked, can we learn a good estimator which doesn't rely on a training set with the correct recombination rate? So here we have 4 adaptive neural networks trained on different data sets. One is for without recombination, with intermediate recombination. The pink one is for high recombination. And then we have a red one. The red one is trained with various different recombination rates in the data sets. And then even this one hidden layer neural network is then able to approximate the optimal estimator for different recombination rates. So for low recombination rates, Fuchik's estimator is best, and the adaptive neural network can learn that. And for high recombination rates, Watterson is the best, and the neural network also learns that. And for intermediate, it's also superior to the other ones. In contrast, if you train it on the data set where only, for example, high recombination rate is available, then it's of course much worse if there's no recombination. Okay, so far for our starting in this area. What we're trying to do within the next years is to improve effective simulation of bacterial genome evolution, because so far MS Prime is mainly focusing on human genome evolution. We want to understand how machine learning algorithms can leverage the phylogenetic information which is contained implicitly in the data, and to finally develop phylogeny-aware machine learning tools. And we're also working a little bit on unsupervised feature selection in forensic classification, which I didn't talk at all about today, but I just wanted to mention it quickly here. So take-home messages would be the inherent dependency in population genetic data can be approximated by trees, and this information can be very valuable. Machine learning in population genetic relies heavily on simulated training data and also their assumptions, and there's no easy way around that. And theoretical results can be used to improve the performance of neural networks in PopGen, as we've done in this simple study I just showed you. And with that, I would like to thank you for your attention, and my collaborators, Peter, Clara, who is also in Tübingen now, Richard, away, and of course Jerome.